Welcome to Weekly Neurology Deep Dive. This podcast is for educational purposes only and has no commercial intent.
Good to be here.
Today we're diving into the complex world of Alzheimer's disease genetics. We'll be guided by a recent review article,
Yes.
The Spectrum of Genetic Risk in Alzheimer's Disease by Karagas et al.
Right.
It was published in Neurologynet 2025. We'll be providing you with an overview, but we strongly encourage you to read the full paper for a complete understanding.
Absolutely. It's a field with direct implications for neurologists, in their daily practice.
Indeed. Let's distinguish between the two main types of Alzheimer's.
Okay.
Early onset and late onset.
Right. So early onset familial Alzheimer's disease, or EOFAD,
EOFAD.
is less common, affecting less than 1% of Alzheimer's patients.
Okay.
It typically appears before age 65 and has a strong genetic component.
Right.
Late onset Alzheimer's disease, or LOAD, is the more prevalent form affecting those 65 and older.
Okay.
The genetic picture here is more intricate.
Involving multiple genes and environmental factors.
So EOFAD is like a single gene taking center stage,
Yeah.
while LOAD involves a complex interplay of genetic and environmental influences.
Exactly.
How does that translate to clinical practice for neurologists?
Well, it's a crucial distinction for EOFAD. Identifying the specific gene mutation can guide family counseling and potentially inform future treatment strategies.
Okay.
For LOAD, it's more about understanding a patient's overall risk profile.
I see. So it's really about sort of predicting the future in a sense.
In a way, yes.
Okay. Let's delve into the genes responsible for EOFAD.
Sure.
What are the main culprits?
Mutations in 3 genes account for the majority of EOFAD cases.
Okay.
APP, PSEN1, and PSEN2.
And these genes are involved in?
These genes are involved in the production of amyloid 42, a protein that forms amyloid plaques,
Right.
a hallmark of Alzheimer's.
Hallmark.
Mutations disrupt this process, leading to an overproduction of amyloid 42. So these mutations essentially guarantee the development of Alzheimer's.
Yes. These mutations are highly penetrant, meaning individuals who inherit them have a very high likelihood of developing the disease.
The link between EOFAD and Down syndrome is fascinating.
It is.
Can you elaborate on that connection?
The APP gene is located on chromosome 21, the same chromosome that's triplicated in Down syndrome.
Right.
This observation led to early insights about APP's role in Alzheimer's.
Interesting.
Individuals with Down syndrome have a significantly increased risk of developing early onset Alzheimer's, often in their 40s or 50s.
So a chromosomal abnormality in Down syndrome provided a crucial clue to understanding the genetic basis of EOFAD.
Precisely.
Moving on to LOAD, the APOE gene, particularly the E4 allele, seems to play a key role.
The APOE E4 allele is the strongest genetic risk factor for late-onset Alzheimer's.
Yeah.
A large multi-ethnic meta-analysis found individuals with the E4 allele have a 3.68-fold increased risk of developing Alzheimer's.
So that's a substantial increase. What are the implications of that for neurologists?
It's important for a few reasons. Knowing a patient's APOE genotype can inform your clinical assessment and management decisions.
Okay.
For instance, consider genotyping before prescribing anti-amyloid therapies like lecanemab, as E4 carriers show greater vulnerability to ARA, amyloid-related imaging abnormalities.
I see. That's a very practical application of this genetic information.
Absolutely.
Does the number of copies of the E4 allele a person inherits affect their risk?
Yes. The E4 allele demonstrates a gene dose effect.
Dose effect.
1 copy increases risk, but 2 copies dramatically amplify it.
Okay.
For example, compared to the common E3-E3 genotype, the risk of AD is 2-4-fold higher with either the E2-E4 or E3-E4 genotype.
Interesting. So understanding a patient's APOE genotype can help predict their risk and tailor treatment strategies.
Precisely. Interestingly, there's also the APOE E2 allele, which has a protective effect.
So some variations of the APOE gene increase risk, while others offer protection.
Studies suggest the E2 allele offers about a 40% lower risk compared to the common E3-E3 genotype.
Okay.
For instance, compared to the common E3-E3 genotype, the risk of AD is 40% lower with either the E2-E3 and E2-E2 genotypes.
Okay.
However, this protective effect seems to vary across different populations, particularly in non-European cohorts.
So this highlights the importance of considering a patient's ethnicity and genetic background when assessing their risk.
Yes, it's not a one-size-fits-all approach.
Right.
The E4 allele consistently increases AD risk,
Mm-hmm.
but E2's protective effect is less clear-cut,
Yeah.
especially across diverse populations.
The APOE gene is clearly a major player in the AD landscape.
It is.
Does its influence extend beyond its direct effect on risk?
Absolutely. APOE genotypes can dramatically alter the effect size estimates of other genetic variants.
That's interesting.
This complexity underscores the need for comprehensive genetic analysis, not just focusing on APOE in isolation.
So while APOE is a powerful predictor, it's just one piece of a larger genetic puzzle.
Exactly. There are other genes like SORL1 and TREM2 that are also emerging as significant contributors to AD risk.
What makes these genes noteworthy?
Well, both SORL1 and TREM2 were initially discovered through different avenues of research, but now we recognize their roles in Alzheimer's.
Okay.
SORL1 is involved in the endocytic pathway, crucial for clearing amyloid from the brain.
Right.
Mutations in ROL1 disrupt this clearing process, potentially contributing to amyloid buildup.
Okay.
TREM2, on the other hand, is linked to microglial function and neuroinflammation. Variants in TREM2 appear to impair the brain's immune response, which may accelerate AD progression.
It seems like with every new discovery, our understanding of the genetic underpinnings of Alzheimer's becomes more nuanced.
That's the nature of scientific progress.
Right.
As we uncover more genes and their functions, we gain a deeper understanding of the disease's complexity.
But even with all these identified genes,
we
still haven't accounted for the full heritability of Alzheimer's.
You're referring to the concept of missing heritability.
Yes, missing heritability.
Twin studies estimate the heritability of Alzheimer's to be between 58% and 79%.
Wow.
This means genetic factors account for a large portion of the risk.
Right.
However, the genes we've identified so far only explain a fraction of that.
So there's still much more to uncover. How do we approach this missing heritability?
Genome-wide association studies, or GWAS, are playing a critical role in this ongoing detective work.
GWAS.
These studies have identified numerous common variants linked to late-onset Alzheimer's.
And each of these variants might only have a small individual effect.
Yes.
But collectively, they contribute to the overall risk.
Precisely. GWAS has revealed intriguing genes like ABCA7 and BIN1.
Okay.
ABCA7 seems to be involved in lipid metabolism and the transport of amyloid across cell membranes. BIN1 plays a role in endocytosis, the process crucial for cells to internalize molecules, including amyloid board.
So while individually these variants may have subtle effects, understanding their collective contribution is vital for a comprehensive picture of Alzheimer's risk.
Yes, and that's where polygenic risk scores, or PRSs, come into play.
PRSs, can you explain what PRSs are and how they're being used in Alzheimer's research?
PRSs aim to assess an individual's genetic risk by combining information from multiple variants.
Okay.
It's a way to move beyond looking at a single gene and consider the cumulative impact of numerous genetic factors on a person's likelihood of developing Alzheimer's.
So PRSs provide a more holistic view of the genetic landscape.
Precisely. They hold immense potential for clinical practice.
Okay.
Imagine identifying individuals at high risk for Alzheimer's before they even show symptoms. This could revolutionize how we approach early intervention and prevention.
That sounds promising, but are PRSs ready for prime time? What are the challenges involved in their clinical application?
There are still challenges to overcome.
Okay.
One significant challenge is the outsized influence of APOE. The E4 allele's strong effect can overshadow the contributions of other variants in a PRS.
So APOE's dominance makes it difficult to accurately assess the combined impact of other variants. Are there any other limitations?
Another challenge is the lack of representation from diverse populations in the data used to develop PRSs.
Okay.
This limits their applicability to individuals from non-European backgrounds, a major concern.
Right.
Despite these challenges, there has been some progress. Studies have shown individuals with higher PRSs tend to have an earlier onset of Alzheimer's and a higher incidence of the disease.
So PRSs, while still under development, show promise for identifying individuals at risk and potentially guiding treatment decisions.
Further research is needed to refine their accuracy, address the issue of APOE's dominance, and ensure they are inclusive and applicable to individuals from diverse backgrounds.
Welcome back to Weekly Neurology Deep Dive. As we've seen, the field of Alzheimer's genetics is constantly evolving, offering new insights into the disease's complexity and revealing potential avenues for targeted therapies. These advances directly impact how neurologists approach diagnosis, treatment, and prevention.
Right.
Staying abreast of these discoveries is essential for providing, optimal care to our patients.
It is.
So let's revisit those common variants identified through GWS.
Okay.
You mentioned ABCA7 and BEON1 earlier.
Right.
Could you elaborate on their clinical relevance?
Sure. ABCA7, involved in lipid metabolism and amyloid transport, presents a promising target for therapeutic intervention.
Okay.
By understanding its role in amyloid clearance,
Right.
researchers are exploring ways to enhance this process and potentially reduce amyloid buildup.
So understanding ABCA7's function could lead to the development of new drugs that target amyloid clearance pathways.
Exactly. Then there's BEON1 involved in endocytosis, a process of cells internalizing molecules, including amyloid. Disruptions in BEON1 could hinder this process, contributing to the accumulation of amyloid plaques.
Okay.
Further research into BEON1's function could reveal new targets for enhancing amyloid clearance.
It seems like each gene we uncover adds another layer to our understanding of the intricate mechanisms involved in Alzheimer's development.
It's like assembling a complex puzzle.
Right.
And each gene provides a vital piece.
A piece.
However, the mystery of missing heritability remains.
Right.
While GWAs has identified numerous common variants, there's still a significant portion of Alzheimer's heritability we haven't accounted for.
So how do we tackle this challenge and uncover the remaining genetic contributors?
Well, research into rare variants is crucial.
Rare variants.
These variations occurring in less than 1% of the population can have a substantial impact on disease risk.
Right.
Next-generation sequencing has revolutionized our ability to identify these rare variants, providing deeper insights into the pathways involved in Alzheimer's and potential targets for therapeutic intervention.
So rare variants, though less frequent, may hold valuable clues to understanding the disease's genetic underpinnings.
Yes.
Could you give some examples of these rare variants and their implications?
We've discussed SORL1 and TREM2, but many others are being investigated. For instance, mutations in the NOTCH3 gene have been linked to a rare form of dementia called CADASIL, characterized by stroke and vascular dementia.
Interesting.
Interestingly, some NOTCH3 variants have also been associated with an increased risk of Alzheimer's, suggesting potential overlaps in disease pathways.
These connections between seemingly distinct dementias highlight the complexity of neurodegenerative diseases.
Indeed. It's a reminder that these diseases may share common underlying mechanisms.
Right.
And understanding these shared pathways could lead to broader therapeutic applications.
Let's return to polygenic risk scores. Given their potential for early risk assessment, are there any specific ways neurologists can incorporate PRSs into their clinical practice today?
While PRSs are still under development, neurologists can use them to educate patients about their genetic risk factors and encourage lifestyle modifications that may mitigate that risk.
Right.
For example, individuals with a high PRS may benefit from more aggressive management of cardiovascular health factors, given the established link between vascular health and cognitive decline.
So even though PRSs aren't yet a definitive diagnostic tool, they can be valuable for patient education and guiding preventative measures.
Precisely. It's about empowering patients with knowledge and encouraging proactive steps to maintain brain health.
Earlier, you mentioned the outsized influence of APOE on PRSs.
Yes.
Are researchers making progress in addressing this challenge?
They are developing algorithms that adjust for APOE's effect, allowing for a more balanced assessment of other contributing variants. Another area of active research is improving the representation of diverse populations in genetic datasets used to develop PRSs, ensuring their applicability across different ancestries.
This focus on inclusivity is crucial to ensure the benefits of genetic research reach all individuals regardless of their background.
Absolutely. As we enhance the accuracy and inclusivity of PRSs, their utility in clinical practice will undoubtedly grow.
Right.
We can envision a future where PRSs combined with other clinical data help personalize Alzheimer's prevention and treatment strategies.
That's a compelling vision for the future of Alzheimer's care.
It is.
What other advancements on the horizon hold promise for neurologists and their patients?
Well, the discovery of protective genes is fueling research into novel therapeutic approaches.
Protective genes.
Understanding how these genes confer resilience against Alzheimer's could lead to the development of drugs that mimic their protective effects.
So this research into protective genes is like searching for the body's own defenses against the disease.
Precisely. Imagine harnessing the power of these natural protective mechanisms
Right.
to develop therapies that enhance the brain's resilience.
It's a fascinating area of exploration.
It is.
Could you provide some specific examples of research being done in this field?
Researchers are investigating the molecular pathways involved in the protective effects of the KLVS variant, hoping to develop drugs that target those pathways.
Okay.
Studies are also underway to understand how the APOE Christchurch and Erlen Kolbus variants confer their remarkable resilience,
Right.
paving the way for potential therapies that mimic their actions.
These research efforts offer hope for developing entirely new classes of Alzheimer's treatments that go beyond simply targeting amyloid or tau, the traditional hallmarks of the disease.
The research landscape is shifting.
It is.
Instead of focusing solely on what goes wrong in Alzheimer's,
Right.
we're now exploring what goes right in individuals who exhibit resilience.
Okay.
This shift in perspective opens up exciting new avenues for therapeutic development.
This is a hopeful note on which to conclude part 2 of our deep dive. Welcome back to Weekly Neurology Deep Dive. We've covered a vast landscape of Alzheimer's genetics, from the roles of specific genes to the concept of polygenic risk scores, and even protective variants. As we conclude this deep dive, let's shift our focus to the practical and ethical considerations of genetic testing for Alzheimer's risk.
It's a crucial aspect of translating these genetic discoveries into clinical practice.
Right.
Genetic testing for Alzheimer's disease risk is not routinely recommended for everyone.
Right.
Neurologists need to understand when it's appropriate
Okay.
and how to guide patients through this complex decision-making process.
What are the situations where genetic testing for Alzheimer's risk might be considered?
Individuals with a strong family history of early-onset Alzheimer's disease,
Correct.
particularly those with multiple affected relatives, might consider genetic testing.
Right.
It can help them understand their personal risk and make informed decisions about family planning and lifestyle choices. However, it's important to emphasize that genetic testing for Alzheimer's is a complex decision with potential emotional and psychological implications.
So while genetic testing can provide valuable information,
Yes.
it's not a decision to be taken lightly?
No, it's not.
What role does genetic counseling play in this process?
Genetic counseling is essential.
Okay.
It helps individuals understand the risks, benefits, and limitations of testing. A genetic counselor can explain the inheritance patterns of Alzheimer's,
Right.
discuss the potential implications of a positive or negative result,
Okay.
and address any emotional concerns the individual might have.
It sounds like genetic counseling provides crucial support and guidance
It does.
before and after testing. What are some key points neurologists should emphasize when discussing genetic testing with their patients?
Firstly, a positive genetic test doesn't guarantee that someone will develop Alzheimer's.
Right.
It's about understanding probabilities and risk factors. Secondly, genetic testing provides one piece of the puzzle, but not the whole picture.
Right.
Other factors like lifestyle, environment, and overall health also contribute to an individual's risk. Lastly, genetic testing for Alzheimer's can have significant emotional and familial implications.
Yes.
Patients need to be fully informed and prepared for these potential consequences.
So it's about having a balanced and informed conversation with patients,
It is.
ensuring they understand both the potential benefits and limitations of genetic testing.
Precisely. It's about empowering patients to make informed decisions that align with their personal values and circumstances.
Right.
As our understanding of Alzheimer's genetics continues to evolve, we can expect to see more targeted and personalized approaches to risk assessment and prevention.
Okay.
This will further enhance the utility of genetic testing in clinical practice.
It seems like the field of Alzheimer's genetics is rapidly progressing, bringing both exciting possibilities and complex ethical considerations.
Absolutely. The knowledge we're gaining has the potential to transform how we approach Alzheimer's care,
Right.
leading to earlier interventions, personalized treatments, and ultimately more effective prevention strategies.
This has been an incredibly insightful deep dive into the world of Alzheimer's genetics.
It has been a pleasure.
As we wrap up, what key takeaway message would you leave for our listeners who are primarily neurologists seeking to apply this knowledge in their practice?
Genetics is playing an increasingly important role in our understanding and management of Alzheimer's disease. By staying informed about the latest genetic discoveries
Right.
and engaging in thoughtful conversations with our patients about genetic testing, we can harness this knowledge to provide more personalized and effective care.
Thank you for sharing your expertise and for guiding us through this complex and rapidly evolving field. To our listeners, we encourage you to continue exploring the fascinating world of Alzheimer's genetics by embracing lifelong learning and remaining at the forefront of scientific discovery. We can work together to improve the lives of individuals and families affected by this devastating disease. Until next time, keep those brains engaged and those minds curious.